Hi everybody, welcome back. Hope you all enjoyed the first day. So I guess we have a couple talks this morning and I'm starting up. So my name is Camille Scott. I'm a graduate student in Titus Brown's lab at UC Davis, and I'm going to be talking about sequence assembly graphs and their construction. So this is going to be introductory. So I'm trying to aim for a wide audience here. So it's not super technical. There's no eigenvalues in this talk, so you can either relax or maybe that's not so exciting for you. But so who am I and why am I here? Like I just said, I'm at UC Davis in Titus Brown's lab. So generally, I study streaming methods for constructing and analyzing assembly graphs, which this talk is going to get into. In general, our lab has an interest in lots of things to do with assembly graphs, open science. We're really big about teaching computational skills, particularly to graduate students in biology and related fields. And we're big about scientific software sustainability, so we tend to give talks and ramble about testing and open source and packaging and all of this sort of stuff, but I'm not going to really get into that today. And or perhaps why I'm here is either because I was invited or because my PI is currently frolicking around the redwoods with his family. So maybe if that wasn't the case, you'd be hearing from him instead. So, all right, let's get into it then. So the motivating problem here is how do we infer the sequence of symbols representing a molecule of DNA, right? So this is an illustration by David Goodsell at Scripps. He does these really beautiful illustrations of cells. So DNA in a cell is this blob of goop basically all tangled up, and here they've got like a replication fork in the middle here is this little purple blob, and then it's being split apart and packed into chromatin, and it's always important to remember for us bioinformaticians that everything we work with is actually this. It's not just a bunch of strings, which is what we think of things as. And so how do we turn this into those strings though, right? So the problem here is that generally when you observe things, you damage them in some way, right? So if I want to interrogate a molecule, I have to abuse it. I've got to shoot it with lasers or perform chemical reactions, put electrical charge across it, and usually this breaks down the molecule, right? So what this tends to do, especially with the current generation of commonly used sequencing tech, is that this limits the length of sequence that we can observe at one time. So usually that's 100 to 250 bases. There's some advanced platforms, prototype platforms that are being used now that get that all the way up to 100 kilobases with a really high error rate. But regardless, we're limited as to how much we can actually sample from some molecule of DNA. And so what this means is that the big thing that everybody uses currently is shotgun sequencing. So what we do is we randomly sample reads, we call them, from the underlying sequence. So if this bottom bit here is our DNA molecule we're trying to sequence, what we're going to end up with are these overlapping samples, hopefully covering the whole sequence. And there's a whole set of statistics associated with how much sequencing do we need to do, depending on how big the underlying genome is. We call these the Landor-Roach-Waterman statistics. Spoiler alert, it's Poisson distributed. I'm not going to get into the math. But regardless, what this means is that usually we do pretty high coverage, pretty high redundancy to try to guarantee that we cover everything. So 50 to 100x coverage is pretty common for these data sets. And of course, nothing is easy in biology, so the sequencing process has an error rate with shorter reads, which is what I'm going to focus on. That error rate is relatively low, maybe 0.1% to 0.5%, depending on the sample. And with that 100x coverage, and say a genome the size of the human genome, which is 3 gigabases, you end up with maybe 300 gigabytes of reads for just one sample. So it's a ton of string data, right? It's a ton of string data that we're trying to organize and process. So when I refer to sequence assembly, I'm saying we have these reads, we have this giant sample, this big pile of strings, and we want to simplify that random sampling down to some approximation of the underlying sequence it was sampled from. So that's actually the assembly process, right? So, so the first kind of naive way that you would think about doing this gives rise to what we would call the overlap layout consensus approach. And this was the first way that people started doing this. So the basic process is that first you try to find all the overlaps between all of the reads. You put this into a graph, that's the overlap graph. We simplify the graph as much as possible in the layout step. And then when you have conflicting areas, you choose the best option, right? So this is coming up with a consensus. And so these OLC approaches, after you simplified the graph, these were the first assembly graphs, right? So what does this look like? Well, if we had the previous sampling of reads, we're going to find all of these overlaps between them. In this case, I did overlaps up to one base. Obviously, you wouldn't actually use one base at a time. You'd do overlaps of like 20 to 30 bases. But you end up with this large tangled up graph of all the overlaps that now you need to simplify. So a naive approach might just be do an all-by-all alignment between all of the reads, right? But on modern sequencers where you have like 100 million reads or maybe 1 billion reads, maybe metagenome samples, you have like 10 billion reads. This is obviously absurdly impractical. Individual alignment between two sequences is order n squared, more or less. And then if you have order n squared to actually do all those alignments, this would just explode, right? So naturally, in order to help solve this problem, another graph structure comes to the rescue. So that structure is the DeBruyne graph. So DeBruyne graphs started off as this kind of obscure combinatorial problem in the late 1800s. Nobody paid attention to them for a long time until some networking applications showed up, like in the 90s, and then eventually genome assembly. So the kind of basic mathematical definition of a complete DeBruyne graph is that a DeBruyne graph is of order k over some alphabet, sigma, and then that DeBruyne graph is over all possible sequences of length k over that alphabet. And so we draw an edge between two vertices if the length k minus 1 suffix of the first vertice is the same as the length k minus 1 prefix of the other vertex, right? So for example, here we have a complete DeBruyne graph of order k equals 3 just over the alphabet 0, 1, right? So there's some interesting properties of these. For example, the complete DeBruyne graph is Eulerian, so it has a path hitting every edge exactly once, and Hamiltonian, so it has a path hitting every node exactly once. So you can always traverse everything in this graph. As an aside, fun fact, this is one reason why when you're entering key codes, like to get into your storage unit, you usually have to put a sentinel character at the end, because if you go and walk this graph, you'll extract. If you walk every node in the graph, you'll get a DeBruyne sequence. So DeBruyne sequence contains all of the possible k-mers. And if you type the DeBruyne sequence into the keypad, you'll be able to break into it. You'll be able to brute force it in a lot less steps than if you just had to put the sentinel after. So if you're trying to break into something, just keep that in mind, but don't credit me. So DeBruyne graphs have this really convenient property, you might notice, that the edges are implicitly defined, right? So if I have some vertex, I can just go and find all of its neighbors by taking I can find all of its out neighbors, right? Its right neighbors by taking the suffix, and then just adding each base and each symbol in the alphabet to it and querying, right? And I can do the same for the in neighbors as well. And usually, like in our use case here, our alphabet's going to be, it's just going to be 4 symbols, A, C, G, and T. So that's pretty efficient, right? I only have to check 4 things. So all I actually need is a big set. I just need a big bag of nodes, and I have a DeBruyne graph implicitly defined. And so, for example, for DNA, if I had a sequence of length 50, this would be all of the 21 mers from that, right? So really all you have to do to get the k-mers, we call them, right? To get the k-mers from this sequence is just iterate a sliding window down the sequence. And what this means is that I can build a DeBruyne graph by just iterating through every sequence, breaking it into k-mers and sticking them in a bag, right? Sticking them in a set. And so that's much more convenient and easy than trying to do all-by-all alignment between the reads, because this basically means that I get every k-1 overlap for free, right? I don't have to actually do any alignment whatsoever. And fixed length strings are nice because we can hash them, right? They're very fast to process generally, so this suddenly is very convenient. So for example, for DNA, for k equals 3, these are a couple of isomorphic DeBruyne graphs that just have different underlying sequences, right? And you can see some bifurcations in these. Okay, so as I said, that implicit edge property means that we solve that major problem for free. We get all overlaps of length k-1. So k is this dominating parameter for working with DeBruyne graphs and for k-mers in general. So if you're working with bioinformatics, computational biology, you see this property k in all sorts of different software, because we don't just use this for assembly, right? We use this for aligners, for seeding into string alignments. We use it for all sorts of weird non-assembly statistics, right? And this k is everywhere. So the big thing for DeBruyne graphs and assembly graphs is that if we choose k to be too small, right, we're not going to uniquely index different areas of the graph, right? So if I choose like k equals 10 in the human genome, there's going to be like hundreds of millions of 10-mers that are, non-unique, right? But if I choose it too large, and my error rate is like 0.5%, if I choose k equals like 100, and my reads are 150, right? Suddenly I'm only going to have like 50 k-mers per read, and it's really likely that every single k-mer intersects an error, right? So now the whole graph becomes disconnected. So you need to find this optimal k size for assembling a particular genome. And so another property of these, which can either be a good thing or a bad thing, is that every time you have a point mutation or an error, you're going to add k new vertices to the graph. So if you're interested in finding single nucleotide polymorphisms, right, so point mutations, this is a useful property because it means I can just go through the graph and look for bubbles. That's what we call them. If I have a really erroneous data set, this becomes a big problem because it means every time I have an error, I'm adding k new nodes to the graph, and so memory tends to explode. So the trade-off for DeBruyne graphs is that they tend to use a ton of memory unless you come up with efficient data structures. So they're really easy to grok. They're really easy to implement. You can do it. You can build a DeBruyne graph in like 20 lines of Python. You won't be able to do anything with it, but you can build it, right? But like you have this poor memory scaling. And then if you have you'll get the bubble if the error is in the middle of the read. If it's at the end of the read, right, you'll get a little tip. So you won't have the complete bubble. You'll just have a little hair sticking off the DeBruyne graph. Right, so after we build this raw DeBruyne graph, right, this thing is huge, and it's going to have a bunch of conflicting areas. It's going to be really messy. So we have to do a series of cleaning operations. So the common ones are bubble popping. So the trick here being that we want to pop the bubbles that come from errors, but we don't want to pop the bubbles that come from actual mutations. If we have like a diploid genome or like something like hexaploidal, which is really horrible for this. But so we want to try to figure out which ones are good bubbles, which ones are bad bubbles. We'll do tip removal, right? We'll start doing things like contracting linear paths, which I'll get into in a little bit. So for example, we have here is from an early DeBruyne graph assembler called Velvet. And this was a tour bus algorithm. So this was this iterative method for collapsing redundant paths in the graph, right? And there's a whole series of heuristics for this because the thing that I did not tell you is that this problem actually is NP complete. Actually doing optimally assembling out all of the original sequence is NP complete. So we do a bunch of heuristics and pretend that we're actually getting really good results. Right, so that contracting linear paths property is important because this gives rise to another sort of DeBruyne graph that we commonly use, and this is called the compact DeBruyne graph. So this is relatively simple conceptually. It just means when we have these long chains of k-mers, right? And so I didn't actually draw the sequence on here, but these could be k-mers of any size, right? Overlapping subsequences. So when we have these long chains with no branch points, so if the in degree or the out degree is 1, then we can collapse that down into one node, right? So we'll just make it into one big node and store the string. And so this gives us the compact DeBruyne graph. So the linear paths can either be called contigs or unitigs, depending on who you ask. I usually call them unitigs because contigs is more generalized. But these directly represent portions of the underlying sequence. So if I wanted a really basic genome assembler, I could just build the DeBruyne graph, like cut the tips off, compress the linear paths, and just spit those out. Right? So it wouldn't be a very good assembly because there's going to be all of these branch points, but it would be an assembly. So actual assemblers, of course, do more than this, right? So they're usually going to perform additional heuristics to try to actually span across these high degree nodes, so these nodes of greater than degree 2. Of course, we call them high degree nodes in the biz, but we actually can have only up to degree 8. So for normal graph theorists, these are like not high degree at all. These are relatively simple nodes. But so to give you an example of what these underlying assembly graphs end up looking like, this is an assembly graph generated with k equals 51 from a Salmonella genome. So this was made with a program called Bandage that you can find on GitHub. I didn't write it, but it's a wonderful little utility for looking at these. So Salmonella is like not a very big genome, right? And like this is already a big hairball. So this is an example of why the k size really matters. If I kick the k size up to 61, we can see that it becomes a lot more comprehensible. And these long chains are gonna directly correspond to complete sequences that we can spit out, right, and do further analysis on. Of course, if I choose k equals 91, I just completely destroy the graph because there's too many errors, and so the graph becomes super disconnected. So where does all of this graph complexity come from other than errors, right? So we can imagine these technical errors from the sequencing instrument, but it would, you imagine like, isn't a genome just a big pile of chromosomes, right? It should be like, 10, 20 chromosomes, you spit out 10, 20 contigs, right? Well, unfortunately, genomes are filled with all sorts of complexity. Namely, they have a lot of repetitive regions, right? So you can have these simpler constructs like tandem repeats. So this might be like, if the red might be something like a gene, like, some gene, and then maybe the next 2 red bits are like it got tandemly duplicated during the cell division process. Maybe eventually during evolution, those degrade into pseudogenes. And in the end, they get separated by some other sequence, right? And when I have a DeBruyne graph, like this all gets compacted down into like these repetitive regions because we're just using k-mers, right? Get compacted down into single nodes. And so you end up with these complex repeat structures. You might also have situations like this is like basically the most complex situation you can get when you sequence messenger RNA. So this is what they call like a intertranscript circular repeat. And that terminology comes from the paper for an assembler called Shannon. So you end up with these complex structures like this, right? That are going to be difficult to actually deal with. So oftentimes it isn't actually possible to output a faithful representation of the underlying genome. So like in this case, right, if these are my coverages, so how many times did I observe a particular k-mer? It's going to mean how many reads did it show up in. I can make a good guess that if I start down here in the lower left, traverse through this middle node, I can go to the upper right, right? But if I come in at any of these other points, I can't really make a good guess as to where to go. So I have this ambiguity that I can't resolve. So increasingly, the field is moving towards the idea that maybe we just shouldn't try to output flat sequences. Maybe we should just leave things in the graph, right? So motivating that is like, what do we do with the data traditionally? So usually these analysis methods start, something like we have a critter, we stick it in a blender, right? We sequence the DNA, we assemble it, we spit out a whole pile of disconnected sequences, contigs, right? And then we do some stuff to it. So we do like variant calling or gene prediction or copy number analysis. And a bunch of these things like variant calling mean that now we go back to the beginning and we take the reads, take the original reads, and we like map them back to the thing we assembled, right? And then and then after that, we do our alignment, but then we have to like convert it to a different format. And then we have to convert it to another format. And then we have to filter it. And then we have to sort the resulting coordinates. And then we can finally start like variant calling. So like the most commonly used alignment pipeline that you use for variant calling ends up looking over all of the reads like 4 to 5 times, right? So and that's ridiculous when you have like hundreds of gigabytes. So some modern methods are to, say, call the variance directly on the DeBruyne graph, right? So with a little bit of auxiliary data, I can put multiple samples into the same DeBruyne graph and mark where they came from, right? So this comes from the Cortex assembler. This is by Zam and Iqbal. And they introduced what they called colored DeBruyne graphs. I like to argue with him on Twitter because they're not colored graphs. I should call them labeled graphs. Graph coloring already is a thing. But regardless, the point is that you can associate a color with the original sample that it came from, so maybe the original individual. And then you can go through and you can start calling the variance just directly on the graph by looking for bubbles or by looking for particular structural variations like transpositions are going to have a particular signature in the graph. And this totally short circuits this whole like alignment step, sorting things, looking at the reads a half a dozen times. Now I can just load it into the graph and simplify the graph a bit, which I had to do anyway during the assembly process, and just do the process directly on the DeBruyne graph. Or, for example, alignment. So a common task is that I have a bunch of reads, or I have some genes or something, and I want to align them to a reference genome. There's been a lot of work recently on aligning directly to graphs. So you can use like a partial order alignment and take a string, a sequence, and actually align directly to the graph. And you never have to actually output any sequence to do this, right? So you can just skip this assembly step entirely. So I have more if you want to hear more, but I think I'm out of time. So the conclusion here is that graphs are great for genomics. We already use them a lot, but let's use them even more, right? So some acknowledgments, my PI, people in my lab, so on and so forth. And I have some links to like our lab website and my website and GitHub and all of that. So thank you. Any questions? Mm-hmm.
So when you're doing the alignment over graphs, you have these variations, and you're saying that you're not able to uniquely represent the underlying DNA. But can you think of that as a probability distribution over what, like a Bayesian sort of, you are uncertain about what the actual underlying DNA is? And so every time you have one of those forks, or every repeat loop or whatever, is that an accurate way to think about this, or am I misplaced?
Yeah, so that's a method that's used. This sort of there's actually a paper that's specifically like Bayesian Bayesian genome assembly. That's more commonly used for transcriptome assembly. So with the transcriptome, instead of sequencing the DNA, we're sequencing the expressed messenger RNAs. So we're going to have variable coverage through transcripts. And in particular, if you want to tease apart alternative splice variants of the same transcript, so you have a couple exons and they're put together in different ways, but always in the same order, right? But maybe they're missing particular exons in the middle. You end up with this little splice. Do I have a picture of it somewhere? No, I don't have one. Should have put one at the end. So you end up with this splice graph, right? And because you have this variable coverage between transcripts, you can use some Bayesian statistics to actually figure out how to cross those junctions. So that is a method that's used, yes.